In terms of regulation, though, chances are it will be illegal for them to require it. But they'll probably be able to accept it. And this technology will allow them to actually quantify the impact of each of these variables on duration of life, including, by the way, blood chemistry. We're going to have all the blood chemistries in there as well once the technology is done. It should be done by next March. Yes?
This isn't on the technology, but just a comment that the statement that you're not going to extend human longevity is giving NASA fits on trying to plan for going to the next star.
Well, it's funny you should mention that, because some of the work that my colleague Bruce Carnes is doing has been funded by NASA. And NASA is working very hard to try to send somebody to Mars. Part of the problem with doing so is that the astronauts that go will be exposed to fairly high levels of radiation along the way. And so there are some compounds that are out there, one of which is called WR2721. This compound, when introduced into the body, seems to protect nuclear DNA from problems associated with radiation exposure. The secondary effects, and I'm not sure if you're aware of this, Rafa, but this particular compound seems to have a side effect. And the side effect appears to be decelerated aging. Not a bad side effect, a good side effect. And so there are a number of compounds, and I'm sure you're going to talk about this, that are out there now that researchers have just discovered that seem to have really good side effects. Normally we always think about bad side effects, but good side effects may be the direction that we're headed with some of these compounds. And I think that probably leads perfectly into the next presentation by Rafa.
Yes,
I
think this has been grand. And when someone says that you look good or you look young, they may be telling you the truth. Would you join me in thanking Jay Olshansky? We have time for about a 10, 15-minute break. So I think there's a coffee refresh. Restrooms are this way. Thank you. Thank you. It's my pleasure to move us to the next session. And I want to again acknowledge the Department of Nutrition Science for their co-sponsorship of this symposium. And it's my pleasure to let Connie Weaver, who is head of that department, introduce our next speaker. While she's here, I just want to say that back in the early 90s when we were creating the gerontology program, sometimes you need people that will just support, an idea or a vision. Connie was one of those people that supported our vision. Thank you, Connie.
Well, thank you, Ken, for all you did to implement the vision. And look at the fun event that you're hosting today. That last speaker was so enjoyable. And when he was talking about we really have two ways to approach single disease or disorder of the past versus multiple organs, I thought his background was leading us to say we've got to approach it differently, not cure, but prevention. And so our next speaker is going to bring prevention, at least from diet perspective. What do we know and what do we don't know? And there's a lot more in the column of what we don't know. I think Rafael de Cabo, who's coming up, will agree with that. But he's right in the middle of finding out some of the things that we know, and we're going to be exploring for a role, because we all want to live longer, healthier, even if we don't increase our longevity. To have a higher quality of life right up, it is certainly an important goal. Well, Rafa was here for his PhD. He studied basic cell and molecular biology in Spain up through a master's degree, and he brought a lot of Spain to Purdue when he was here working on his PhD. So I have to tell you, he was a teaching assistant for a beginning foods lab, and Rafa in Rafa style does a lot more in every aspect of his life, then what are the minimum requirements? So once a semester at the end, he would have the halls full of a big buffet from Spain. So I was introduced to paella and many foods I can still remember because the class ended up serving the department and guests. He did things in a big way. And I finally got to go to Granada, Spain for a meeting last September and could order with some confidence the food, and it was so enjoyable. We're lucky to have attracted him to stay for his career in the U.S. because Spain is a really worthwhile destination. Well, after he finished his PhD in nutrition science, he went on to do a postdoc at National Institute of Aging, and apparently he liked it because he stayed where he is now a senior scientist in the Institute of Aging. He's senior investigator, Laboratory of Experimental Gerontology, National Institute of Aging. He has done so much in the short time since he got his PhD in 2000. He's an editor on 6 different journals. Imagine that. He has received in 2012 the Polly Glenn Award for research on the basic biology of aging. He's a fellow in the Gerontological Society of America, elected member of the Board of Directors, American Aging Association. He's a permanent member of the National Institute of Aging Studies section, which is a really complimentary post for peer review of grants. Rafa, we are so proud of you. Welcome back.
Guys, my pleasure to be here. This room means a lot to me. So back door over there, I defended my thesis about 14 years ago. It was, my committee, and I was having a horrible time, for a while trying to figure out what these guys were going to ask me and they were going to pass me or not. But anyway, so I did pass, and I moved on, and I went to the National Institute on Aging, where I got interested on aging and aging interventions to slow down these particular processes. So this is the picture. I said, we did not plan this, but this is my family. So this is my grandmother, my mom, my sister, and a niece. And this picture I always use to illustrate why studying aging is important, why we all age differently, and why we do see aging in a different way. If we were around the room and we were asking the question, what is aging, to each one of you probably will give me a very personal view of what you think aging is. The same thing that if I ask you, how are you aging? Or what are the causes of aging? You will give me your personal experience. And then the same thing goes with, can we manipulate these aging processes? Can we do anything to slow down or to prevent the aging process? And one thing that this picture also depicts is these women do share culture and environment, genetic information, and they have been educated exactly in the same way. Even with all that apparent homogeneity, none of them are going to die exactly at the same age, exactly the same diseases, and they are not going to go through the life course exactly in the same way that my grandmother did. So what is important to study aging? As Jay mentioned, we are on a brink of a revolution where we're going to have more people on the face of Earth than they are over the age of 65 than those that they are over the age of 5. And the amount of people that they're going to live over the age of 100 years is going to grow tremendously. You're going to quadruple by 2030. And so what is aging important? Jay mentioned this quite a bit. Is aging is the major driving force behind all chronic diseases. The only thing that you need to do probably to get one of these diseases, the 10 killers in the US, is to live long enough. And very likely, you will not just get one of them. If you live long enough, you will accumulate a collection of all of those. So, as Jay said, yes. Well, I mean, some of us are prone to get a collection of them, either by genetics or by diet. And as Jay very well said, if you are able to cure cancer completely, what it will be next on your list of diseases. So we are all not predestined, but we do have a sequence of events where your organism and your organs in your system is going to start shutting down. It's very much like a car. You're going to start, missing a plug. And then the next thing you go is the air filter, and then, it's like, one thing leads to the other. So, but if you're able to fit the plug, then the next thing that's going to break probably is going to be the transmission. If you fit the transmission, then it's going to be the brakes. So, I think that what we're trying to do, what we're trying to understand is, can we, by any means, move the onset of these diseases to the right? Can we compress morbidity and have longer and healthier lives? So the question that we always get asked is, can we stop aging? Can we stop the process going from here to here? And the answer is no. There's nothing right now that we can do to stop the aging process or the aging processes. In laboratory animals, we have been demonstrating over more than a century that if you restrict the total caloric intake of any species by 30 to 40%, you will increase both the mean and the maximum lifespan. And this is normally also associated with a postponement of the onset of more diseases. And as Jay said, one of the things that happen to CR animals is that you also postpone the onset of sexual maturity and also postpone the decline of sexual maturity. These black dots represent when animals, and this is mice, get tumors spontaneously. And you will see that these black circles are representative of the control groups, the open ones from the restricted animals. So when you are on caloric restriction, you will get the incidence of tumors goes down, and not only that, but the amount of tumors per animal also declines. And this is true even if you are exposed, for example, to an induced cancer. So you have a melanoma that has been injected subcutaneously, the 40% restricted animals are able to maintain, repress the growth of that particular tumor. It is not just disease, also function. So if you look at the muscle of a 23-month-old animal, in the ultrastructure, it doesn't look as nice and beautiful as the ultrastructure of the muscle of a younger creature. So caloric restriction throughout the lifespan maintains both health and function. So that was the dogma when I studied in 2000, was in the field, that caloric restriction works absolutely always, regardless of the context, as long as you maintain a good and balanced nutrition. So, and what is important to have a model that you can see how aging is delayed. And the reason is because if we are able to figure out what are the mechanisms behind the aging process, the CR, we will probably provide clues of how the aging process is actually happening and how can we slow it down. And more importantly, we can identify targets that then we can use compounds or interventions that will affect them at the cellular level. Yes?
When you talk about caloric
restriction.
We have so much processed foods in this country. If you look perhaps at another culture where the foods are all natural, say, / organic, you see a significant difference?
So is a very good question. So I think that the more processed the food is, the worse outcomes that it has in terms of health. If you look at specific populations in the world, there's two clear examples. One is a population in Sardinia and a population in Okinawa, that they have excellent diets in general. In both cases, their populations have longer mean lifespans than the mainlands of the respective countries. Okay. So when I got in the field in 2000, around 2000, March 2000, I asked my mentor, so how does caloric restriction work? I mean, it's been known since the beginning of the 19th century. Somebody may have a clue how it got. And he gave me this cartoon. He said, you have caloric restriction, then something happens, and you get this extension in survival. And, since then, in the last 15 years, we have been able to populate that when you decrease total energy intake, there's a whole host of things that happen. So there's something elemental that you're changing when you're affecting the whole entire, metabolism of a particular creature. So my working hypothesis starting very early on was trying to understand how what I'm calling metabolic inputs, and this is the cartoon of a picture, but it could be also a cartoon of an organism, is how you perceive, acquire, and use energy and dispose energy is essential for the aging process. So there's a number of what I call metabolic inputs that are normally perceived and alter what we call bioenergetic sensors. These are highly energetic molecules or pairs, redox pairs, that are able to produce energy within the cell. As soon as you alter these ratios, you're going to have an effect on what we call master regulators of metabolism, that they are specific proteins and molecules that they are capable of seeing how much of each one of these components do you have. And in turn, they will go into the nucleus or into the brain, and they will tell what we call transcription factor coactivators what to do to improve metabolic homeostasis and increase the stress resistance. And those are what we call metabolic outputs. And if those two things normally occur in accordance, then you will have increases in health span and decreases on aging. And those goes in those directions, then you should have increases in longevity. So basically, the hypothesis that I've been testing for the last 14 years is, do we have interventions that enhance the processing and handling of energy will have a positive impact on energy homeostasis, increases stress resistance, and then that leads to improvement in health span and delayed aging. So we're going to go back to this picture. So basically what we're trying to do is to develop interventions that one day we'll be able to see a survival curve, something like this, that you get to the end of your life extremely happy, and one day you just show up dead. And the way that we walk that line is extremely important. I mean, you have two pictures here. This is a Native American from the desert of Arizona at the age of 62. If you did your picture profile in this one, he probably told you that it looks like this one. And this guy is a Zen monk from the Himalayas that probably, have had a much cozier life than the woman on the right. So each one of us will walk through the aging process very differently, and it's going to have to be a compendium of how your genes, the environment, the nutrition, the exercise will affect your survival. So early on, I tried to envision how to test some of the hypotheses. Can I do something in the laboratory to do this? And I developed this aging intervention program. And what I did is I started in a circular way where I took cells and I tried to ask the question, can I do any of these compounds of interventions, if I put them onto these cells, can they have improved responses to stress? Then if I got a positive result, then I could test it in mice or rats, and then these experiments normally take a few days. These experiments take a few years, and then I normally test them then in monkeys, and then we transfer them to humans, and then we look at the whole picture to see if there's any commonality across species on the responses of some of these treatments. So what we try to do is to standardize how we were doing this testing, and this will be this normal diet. Animals, when they are in the laboratory conditions and you have good husbandry technicians, they have extremely cozy lives. They are living in a very controlled and very good environment. So in order to challenge the system, to try to provide a challenge that will occur in a normal setting, I try to give them a high-calorie diet, something like most of the U.S. people will be eating almost daily. And then also have always a positive control that was caloric restriction. One thing that was unique about the design of these experiments was that, different from other studies on longevity done in mice, I did have multiple measurements longitudinally across the whole entire lifespan of a set of parameters that they were not very invasive. That way, at the end of the intervention, I would be able to see if it was health, survival, or both was being affected. So The number of interventions that we have used are both genetic, by targeting a particular gene, and nutritional intervention, by either reducing calories or adding something to the diet or taking away something from the diet, and also environmental, like exercise. And then we look through a number of parameters. We're trying to do this the closest to a human study as it could be. You cannot ask a mouse, how is it feeling, or what is it hurt this morning, but you have a number of assessments that you can do to see how they're doing and how they're feeling. So I'm going to go back to calorie restriction because it was like, this was the hallmark of the field. It was like, an anchoring point that we thought that it was a very extremely solid intervention. But as I went back into the literature and I did my own experiments, there are some progress and some caveats about these experiments. When I started, if you asked in the field what calorie restriction did, it told you that as you reduce progressively the caloric intake of an animal until you reach a point that you cannot reduce it more because then it will be under starvation conditions, you will have a progressive increase both in the mean and maximum lifespan. And that is partly true. But now, just by looking at the literature, we know that genetic background has a tremendous impact. And I'm going to give you a couple of examples. And then how the diet composition also has a tremendous impact. So what I'm going to do for the next few minutes is I'm going to give you a little bit of feedback about that. So in an experiment that they took cousins of mice, so there were multiple families of mice that they were created from a single parental crosses. There was a report that showed that about 42 strains, both males and females, there were about 1/3 of animals that benefited for the effect of calorie restriction, about 1/3 that did not, and importantly, there was 1/3 that actually was detrimental for them. And the very interesting thing is they repeated the experiment in a different place, and the shape of the graph looks identical, but there's an issue with some strains that in this case, they were living longer, and in this case, they're living shorter. So the place where the experiment was conducted had an impact. The outcome of the experiment as a whole did not, where you have some strains that did respond and some of them not. So the point that I'm trying to make here is that the genetic background of the particular individual that you're going to implement calorie restriction may have a tremendous impact in the outcome. So then I looked at this strain. That was a strain that, if you look in the published data, there was mixed reports of responders or no responders to this intervention. So we took a number of animals of this strain and a very commonly used strain. So these are DVA2s and C57s, and we repeated a longitudinal experiment. In this case, one of the things that I did to alter the paradigm was I did a mild calorie restriction 20% or an extreme calorie restriction 40%, both in males and females of both different strains. And this is the body weight trajectories throughout the lifespan of this strain. And you can see immediately that the shapes of these trajectories are very different between males and females of the same strain and across strains. When we look at things like insulin tolerant tests to look how well their glucose homeostasis is, you can see again that the response to the different dosing of calorie restriction is extremely different between females and males and across species. So calorie restriction is not working exactly in the same way across species or across sexes for that matter. Now when we look at survival, that to me was astonishing was that the DVA females, it didn't matter if you were doing 20% or 40%. You live exactly, you had an impact on survival, both mean and maximum lifespan, but you did not have an extra benefit for cutting your calories 40% more. In the DVA males, we did see the initial mortality of the population, but then both 20 and 40% did crossover with increases in mean and maximum lifespan. And the 40% did not really produce much greater benefit in terms of maximum lifespan. Surprise to me was that C57 females, 40% CR didn't do anything. If anything, was detrimental for them. And 20%, this individual here probably is the longest ever lived C57 black 6 that has been reported in the literature. And in the males, you have 20% worked the best, and 40 was also beneficial. So The take-home message here is sex and genetic background play a tremendous role in the impact of this particular intervention, as well as the dose of calorie restriction that you're using. Yes?
We talked about genetic effects.
Mm-hmm.
Is there anything distinctive there?
That's a very good question. So,
I said when you talk about genetic effects, is there anything distinctive
between the two strains.
genetics that would
So the very interesting thing here is both strains are, each one of them individually, are extremely genetic homogeneous. So they are crosses between an original set of parents and then crosses between brothers and sisters. So genetically, they're almost identical. Now, what is also interesting is if you look, they not all die exactly at the same time. So genetics is not just it. It's a stochastic process that takes place that determines when each one of these animals is going to die. Now, are they genetically related? So if you look at the genealogy of the mouse strains, they are very distinct genetically. The black 6 males tend to become obese and develop diabetes by about 9 months of age. These guys do not. Okay? So they do have some traits that differentiate them from each other. So what about diet composition? So if you look back in the literature, and these are papers from 1992. There's a paper, each one of these lines represent about 2,000 animals, and it was done in the Asian Center for Toxicity. And what you can see is just by switching the type of diet that you're giving them, and these are two very well used and very standardized diets, you can see that just the Levitton curve, there is an improvement of survival just by diet alone. And what is very interesting is when you push these animals on calorie restriction, the same amount of calorie restriction, what you see is that if you were in the Marie Morse diet and you were put on calorie restriction, your improvement of survival was 40%. But if you were in NIH 31 diet, you only got a 16% increase. So the diet, the composition of the diet, makes a huge impact on the effect, long-term effect of calorie restriction, as it does in the trajectories of body weights. Now, one of the reasons that I went probably into this field was that when I was a graduate student at Purdue, Rick Weinrock came to give a talk on his calorie restriction monkey colony, and he presented for the first time the data on the macroarray. And I remember that was a very exciting talk. And now and behold, now I'm citing his paper, and I'm working with some of these monkeys. So we've been doing since 1986 a longitudinal study of calorie restriction in rhesus monkeys. That they was studied, one at the University of Wisconsin and the other one in the NIA. What's very interesting is that Wisconsin is seeing that if you look at age-related mortality, they are seeing an increase in survival. And they're also seeing an increase in, now they just published, an increase in the overall mortality. So calorie restriction in the Wisconsin monkeys is having a very beneficial effect. If you look at our paper in Nature a couple of years ago, we do see that the age-related disease almost reached significance, but the overall mortality, we don't see absolutely anything. So in these two studies, caloric restriction is having a tremendous impact on health, but in one case it's having an impact on survival, in the other one it does not. Why? So if you look at the diet composition and the setup of each one of these studies, quite different. So our diet composition differ in a number of metabolites. The feeding regimen that we use in the two colonies is different. The body weight effect, the Wisconsin monkeys tend to be a little bit higher. And the genetic origin of the monkeys, we have either Chinese or Indian monkeys in our colony, and Wisconsin has basically, I think, is Indians. And then the age of onset is very different. Wisconsin studied a large cohort, a middle age, and we have 3 staggered cohorts, a very young, middle age, and very old. Yes? Without altering the micronutrient content. So it's a perfectly balanced diet. It's just a total reduction in calories. No, anyway.
There's been a lot of popularity about fasting.
Yes.
Is that considered in your studies at all, or is that something?
So we're studying fasting, but in a different context. So fasting is a very interesting approach that most religions throughout the world have, ever since I think humanity has promoted long or short period of fasting for the purification of the body.
Like once a week.
Correct. There is an intervention that is also caloric restriction that is every other day feeding or every other day fasting. And now there are a number of interventions that a collaborator of mine, Walter Longo, and I have been working on is like pre-chemo fasting for improving the outcome both of the chemotherapy and decreasing the side effects of the chemotherapy after fasting. So that's, fasting is being used, and we're trying to understand what are the mechanisms behind as well. In progress, correct. And if you think about caloric restriction in the 40%, what we are actually doing, so animals, it depends a lot of the strain, but we're feeding them once a day their food allotment that is 40% less than the L libitum. So what they're doing is normally as soon as you put the food in the cage, they eat it all. So they eat once a day, so they are 24-hour fasting periods.
It's interesting in itself. It's normally 3 times a day or regularly?
Yeah. There's been many studies done on the frequency of dieting. So now, what about pharmacological genetic manipulation that alter the CR response? So remember this cartoon that I put up, and I mentioned SIRT1 as one of the master regulators of metabolism. So actually collaborating with Walter Longo that I just mentioned, we created a SIRT1 knockout animal. So this animal has no SIRT1 protein, and we put them on caloric restriction. This is first the pharmacological intervention. So what we did is try to activate this protein with a particular compound and see how activating this particular protein had an impact on survival. Very quickly, I don't want you to pay too much attention to detail, but what you can see here is when we put the control animals in the blue line in a high-fat, high-sugar diet, we reduce both the mean and the maximal lifespan. When we add this compound, we partly reverse this negative effect on survival of the high-fat diet. And when we implement this compound in the standard diet, we see a squaring of the curve, and it became statistically significant at P01. And there was differences in body weight both in the standard diet and the high-fat diet when you do longitudinally analysis. And there was a slight decrease in the fat mass in the standard diet group. When you look at metabolic parameters, and also markers of health fund, for example, lens opacity, a marker of cataracts, we saw that this compound prevent the progression of cataracts, and the incidence of cataracts in the whole entire population was decreased by about 25%. And the same thing, how were these animals handling glucose? When you challenge them, you can see that glucose, when you give them a bolus of glucose at time 0, they handle glucose so much better. So they were having their diabetic prone phenotype prevented. And when you looked at markers of liver health or overall health, you can see that in most cases, this particular compound was preventing the age effect over the standard diet animals. And you can look at a number of parameters: inflammation, gene expression profiles, so it tells you how each particular tissue is handling this compound. And one thing that you can see is that between the liver and the muscle, there's big differences, just eyeballing at the changes. And we demonstrated as well that this compound was in fact working through SIRT1 using both genetic models and also downstream targets of this particular protein on the acetylation of them and the activity, and that's reflected here. The compound basically, what it did is by increasing the activity of this particular protein, it decreased circulating TNF-alpha, that is a marker of inflammation, LDL, and cholesterol, and it had an effect decreasing cataracts, asynchronous circadian rhythm, decreasing body mass, and a number of benefits in the muscle. And that resulted in changes in health span as survival. So now what happened if we actually, instead of activating this particular pathway, we completely block it by deleting this gene from the mouse? So